Hello, everyone, and welcome to another day of the second wave of the pandemic, science and society. Today, our speaker is Dr. Philip Krause, the Deputy Director of the FDA's Office of Vaccine Research and Review. He is also the chair of the World Health Organization's R&D Blueprint Working Group on COVID-19 vaccines, which worked on the development of a research roadmap for vaccines against SARS-CoV-2. He will be discussing COVID-19 vaccines. Welcome, Dr. Krause.
Thank you very, very much. It's a real pleasure to be here. And so the talk might be more scientific than many of you are accustomed to listening to, but that doesn't mean we can't talk about non-scientific things in the Q&A. And I'll do my best to aim the talk appropriately. Before starting, though, I really want to give thanks to my colleagues at WHO and FDA, as well as members of WHO expert advisory and working groups who have contributed a huge amount of time, energy, and motivation for the ideas that are presented in any talk that I give. But as I stand on the shoulders of giants as I present this work, should I stumble and fall, that is 100% my own fault. And so the views I'm presenting are my own and might or might not represent those of anybody else, much less FDA or WHO. So I just want to start off with some background. What is a vaccine? A vaccine is a product that stimulates a person's immune system to produce immunity to a specific disease, which protects the person from that disease. That definition comes straight from the CDC website. Vaccines are intended to induce an immune response to one or more antigens, where an antigen is some protein usually that is expressed on the surface of a pathogen, where that immune response is expected to provide protection against the consequences of infection by a pathogen that bears the vaccine antigens. And the vaccine antigens can be presented to the immune system in many different ways, so vaccines can have many different forms. And that includes directly giving people proteins to get immune responses to that. It can involve giving people viruses, whether they're live and weakened, whether they've been made so weak that they're replication incompetent, but express some genes anyway, or where they're grown in a cell culture or inactivated, and so they're completely killed. Or as we've learned from the COVID pandemic, there's been a new category of vaccines that has come out, nucleic acid vaccine, which shows a great deal of promise, and I'll talk a little bit more about that. So before I get too deep into my talk, I want to sort of tell you a little bit about some of the players in the vaccine story, because there's a lot of government entities and they all have different responsibilities. So the National Institutes of Health, which is best exemplified by Tony Fauci, who leads the National Institute for Allergy and Infectious Diseases at the NIH, performs and funds research into vaccines. And I've chosen here a picture here that to show you that this is the structure of the spike antigen, which is on the surface of the SARS-CoV-2 virus. And so different types of protein form are depicted in different ways, but this is what the spike looks like when it's on the virus. After it binds to a cell, or after certain things happen to it, it looks very different. And what scientists at the NIH noticed when they were studying not SARS-CoV-2, but MERS, was that if you made some mutations in the spike protein, you could keep it from naturally switching from this pre-fusion format to the post-fusion format. And by making those mutations then, they hypothesized that you would be able to make a better vaccine, because we're trying to protect against a virus that hasn't yet fused with a cell. And because once it's fused with a cell, it's in the cell. And so one of the big findings in the SARS-CoV-2 vaccine story was the making of these mutations that allows the spike to stay in its pre-fusion form. And it's changing just 2 amino acids in this entire very large protein that does that. And this, of course, isn't something that somebody came up with after the COVID epidemic came about. This is something that they were studying beforehand. They were studying it in MERS as well as in some other viruses to understand how it is that these viral proteins change shape when they infect cells. And so this really shows the importance of ongoing basic research and the kind of work that the NIH does. Because without this discovery, we probably would not be in as good a shape as we are with vaccines right now, because almost all of the vaccines currently in use, although not quite all of them, are including these mutations. And the most effective ones, at least as they've been studied in the clinic, all have these mutations. So where does funding for vaccines come from? Vaccine funding has always been a difficult thing because vaccines are given to everybody, and yet these are not highly profitable products. And so over the years, different methods came about for funding them. And maybe 10 years ago or so, the Biomedical Advanced Research and Development Authority was stood up with the idea of funding vaccines, especially against various kinds of biothreat agents. And of course, the Department of Defense funds development of vaccines that are consistent with their mission as well. So a lot of the money comes from these other government agencies. Of course, there are also nonprofit organizations that contribute to the development of certain vaccines. And Operation Warp Speed funded development and pre-purchase of COVID-19 vaccines. And the importance of this can't be overstated, because what this did was it meant that there was a huge amount of money going into not only the development, but also the guarantee for the manufacturers of some of these vaccines that if these vaccines were successful, they would be purchased. And even if they were unsuccessful, they would be purchased. So the companies could proceed along development without taking significant risk to their own bottom lines. And in fact, might even have made some profit, even if their vaccines didn't work for some of the companies. And so what this did was it allowed the companies to skip a lot of the steps that often happen in development. Because usually what is happening in development is a company is constantly asking the question, is it worth continuing this project? Because vaccine development is expensive and the trials are expensive. And so, but by knowing that somebody was going to fund this to the end, they could skip the studies that they were going to do for the purpose of deciding whether it was going to be worth investing further in the vaccine. And they could just concentrate on doing the studies that were necessary to help move the development forward and give us enough evidence that they would be likely safe and effective, and it was reasonable to continue studying them. So without the companies needing to worry about big flops that would have cost their bottom line huge amounts of money, the world, and this is in the US was OWS, but there's been a lot of money going to vaccines from Europe and other places as well, allowed vaccine development to occur at an unprecedented pace without cutting corners in terms of the safety or effectiveness of those vaccines. And the FDA actually also has a little role to play in this, is that if there are products for certain illnesses, and that includes COVID-19, they can give a company a priority review voucher once those products are licensed. Those priority review vouchers can be worth quite a lot of money because they allow a company to come in with another product and get it reviewed more quickly than it would otherwise be reviewed. The Centers for Disease Control address questions of vaccine usage. Actually, Centers for Disease Control and Prevention is their full name. And so this is the vaccine schedule that those of you who are parents or those of you who are kids and saw your parents with these schedules, or perhaps saw these on the wall of your pediatrician, might recognize. But this recommendation of when it is that people should get what vaccine is squarely in the bailiwick of the Advisory Committee for Immunization Practices of the CDC. And so the CDC is giving us this advice on how vaccines should be used in a public health context. So what does FDA do? Well, the FDA is responsible for protecting the public health by ensuring safety, efficacy, and security of human veterinary drugs, biological products, which include vaccines, as well as the other things that are listed there. And so what the FDA does is they provide the initial authorization that allows a company to market a vaccine or allows a vaccine to find its way to people. And the FDA also reviews and regulates all of the testing of the vaccine up until that point where it can be used. So this starts at the beginning with the preclinical animal testing and goes through as the testing goes into humans, and finally through the big phase 3 studies that show that the vaccines are safe and effective. And then the evaluation, which in the case of COVID vaccines has led to emergency use authorizations and might then ultimately also lead to licensure. And the Food and Drug Administration also follows up on these products afterwards to make sure that they're still safe and effective And there that they work together with the CDC and other agencies that are also collecting data on that. So regulatory decision-making considers mostly safety and efficacy, but also actually whether or not the manufacturer can make the product consistently, but that's not on this slide. And so we ask questions like, how effective is good enough? How much evidence of safety is needed? And the regulations and the laws do define standards for this, but the availability of life-saving products relies on science-based decisions that weigh benefit and risk. So challenges in regulating vaccines include that vaccines are given to healthy people, often children, so there's little tolerance for safety issues. Even very safe vaccines can be subject to skepticism, misinformation, and anti-vaccine advocacy. The pre-licensure clinical trials, while they can be quite large, can still only address relatively more common adverse events. Vaccine products are usually pretty complicated, and so you can't really analyze them in the laboratory as closely as you can other kinds of drugs. And so you really have to understand how they're manufactured and make sure that the manufacturing process is consistent. And of course, new technologies are frequently introduced into vaccine evaluation and manufacturing. So there's a need for rapid and informed data-driven decision-making, because in public health threats, of course, we can't wait, or we don't want to wait, but even routine vaccines, if they're beneficial to public health, shouldn't be unnecessarily delayed. This is the challenge that the FDA has. So having set that stage, now let's think about COVID-19 vaccines, where the development really, when this problem approached us, was really quite different from anything the world had ever faced before. Many vaccines were being concurrently developed, which when you have many vaccines being studied at the same time, there's always a risk that just by a statistical fluke, one or more vaccines might appear to work even if it doesn't. On the other hand, we needed multiple vaccines, but at the same time, we were worried that if the first vaccines were weakly effective, that might make it much harder to bring better vaccines out there, because it might be harder to use placebos, and it might be, and they would then need to be compared against weaker vaccines, which would then delay the availability of better vaccines. There was a very specific safety concern with coronaviruses, the so-called enhanced disease. And this was based on an experience that was noted even in the 1960s, where respiratory syncytial virus, which is a virus that infects both children and adults and causes a pneumonia-like or an influenza-like illness, there was a vaccine that was developed for that, and it was given to children who tolerated the vaccine okay, but when they got RSV, they actually got sicker than they would have gotten if they hadn't been vaccinated, than the children who didn't get the vaccine got. And a few children died in these experiments back in the 1960s. And other coronavirus vaccines, at least when studied in animals, seemed to have at least the potential for a similar phenomenon. And so we wanted to be very careful and make sure that the studies addressed adequately the potential risk of enhanced disease. There's a need for reliable data because very high deployment rates were expected initially, and indeed have been achieved. So many people would be immunized before a mistake became apparent. Manufacturing was an issue also because if manufacturing resources were expended on the wrong vaccines, it would impact the ability to make other vaccines. And of course, all of this is this analogy of building an airplane while you're flying it. The vaccine development was occurring in the middle of a pandemic with a high level of scrutiny and a huge desire for rapid results. So as the WHO formulated this more than a year ago, as they met early in the stages of the pandemic, there really were two goals that had to both be met. Confidence in the vaccines, that we're giving the vaccines that would work and would be safe, but also speed. And so this sort of became the mantra of the right vaccines, but promptly. And with, again, the notion that the desire to rapidly deploy the first vaccine shouldn't keep us from finding the right vaccines, and there should be an S at the end of that. One of the things that the WHO did very early in this process was they really brought together the entire scientific community that was interested in COVID vaccine to talk about what needed to be done. And so the plans that the WHO developed were international plans. And one of the things they started off with, and this was suggested actually by one of the manufacturers, is to start off with a statement of collaboration, which representatives of major manufacturers, government agencies, funders, researchers, all signed based on this idea that we were all in this together and we were cooperating, and this was not a situation where people were going to undermine each other. That this is the idea that we were only going to make progress on this disease by being open and transparent and share our data and information in such a way that everybody could make progress more quickly. And so the WHO developed three major areas of focus, and I'm going to talk mostly about the clinical development, but these included animal models. This is a remarkable thing where every week since actually a year ago, January, experts in animal study, as well as the viruses, have been talking about the data that they've been generating. This is people from around the world and presenting the most recent results, even before they could be published, and even before they found their way into some of the archive journals, which would lead to even more rapid publication. And so having this weekly call where this kind of information was being shared really helped keep everybody up to date and help make sure that nobody was wasting time. And very often, suggestions would be made on these calls that would help people move their experiments forward more quickly than they would otherwise have moved forward. So it's sort of like an international lab meeting that made a huge difference. The same thing happened for assay development, because coming up with the right assays to evaluate vaccines especially is a very important critical step. And then something very similar happened for clinical development, including human challenge studies. And we're not going to talk more about human challenge studies, but this idea of whether it was ethical or reasonable to challenge humans with SARS-CoV-2 in order to see, for instance, whether vaccines work was obviously a very contentious one that you probably read in the paper. And if you'd like, we can talk about that in the discussion. But I guess a punchline there is it became difficult to figure out exactly what questions you could answer using those questions, those kinds of models that you couldn't answer as easily using other kinds of approaches. Although there are laboratories now in the United Kingdom that are getting set to start doing some human challenge studies. So the landscape, there are now over 180 entities or 180 vaccines that are out there. Of course, they haven't all made it to the end, but these are at least more than 180 in preclinical development with many different technologies spanning RNA, which is a nucleic acid vaccine, the mRNA vaccine, the modified mRNA, which is what the Moderna and the Pfizer vaccines are, and self-replicating mRNAs. There's DNA, including schemes to get the DNA into cells using a technique called electroporation. Inactivated vaccines, usually with some kind of an adjuvant that is designed to increase the immune response. Live attenuated vaccines, which would grow actually in people and engender immune response that way. Vectored vaccines, where there's some other virus, which isn't the one you're trying to get the immune response to, but it's been engineered so that it expresses an antigen from the pathogen you're trying to protect against. So in most cases would express the SARS-CoV-2 spike antigen. Subunit vaccines, which are proteins that have been produced in cells and are given just as proteins, as well as virus-like particles, which is a kind of a protein vaccine where these proteins assemble to look more like a virus, which is believed to yield a better immune response. We now have 3 deployed in the US under emergency use authorization. And of course, there are others deployed elsewhere in the world under various regulatory mechanisms. And there are 25 vaccines around the world that have reached either phase 3 or phase 2 to 3 trials. So what exactly is the right vaccine? Well, it has to have sufficient efficacy, which means we hope it will last long enough. We definitely want it to be effective against severe disease, because that's what we care about the most, is protect people from going to the hospital and dying. We want it to be effective in relevant subpopulations. If it's shown to be effective only in young people, for instance, that's not going to help the elderly, or perhaps if it's shown only in certain ethnic or racial groups, that's not going to help people in other racial groups, or that you're not going to have as much information about that. And then statistically, it's very important that the precision around the efficacy estimate has to support rational decision-making about vaccine use. And so you don't have to know how to calculate the precision around the estimate of a vaccine effectiveness, but I'll try to give you a flavor of why that's important, because that was one of the big controversies that came up early in vaccine development. It needs to be safe, again, with sufficient duration of follow-up in a large enough population to support deployment in billions of people. It shouldn't cause enhanced disease, and you'd like to have evidence that that's unlikely to happen. And of course, it has to be deployable if you want to use it around the world. You have to be able to manufacture it consistently and at high scale. It has to be stable. It has to be possible to implement in a vaccination program. And there has to be enough of it that can be made. There has to be enough supply that it can be used in many, many people. So when we look at clinical trials at the FDA, there are several basic rules that are essential to assure interpretability of clinical trials. And whenever somebody's in a hurry to develop a vaccine, they sometimes want to break one of these rules. And so I'm just stating them here up front to sort of let you know the kinds of things that the companies need to look at when they do the clinical trials. The trials need to be performed based on good clinical practices, which assure ethics, human subject protections, proper record keeping, and so forth, so that we know that what is found in the trial is something that we can rely on. The success criteria need to be stated in advance and have to follow well-accepted statistical principles. And usually this means a specific result needs to be achieved, along with a corresponding 95% confidence interval that also meets specific criteria. And that's the precision that I was talking about. And then once a trial is started, the circumstances under which you can change the criteria for success are very limited. We've had a number of situations where there have been vaccine manufacturers who wanted to change their success criteria after they started getting results. But of course, once you start getting results, if you then change the rule of the game, then you're breaking some of the basic statistical assumptions that were made when you started the trial. And then, of course, the trial has to collect sufficient data to inform decision-making that requires knowledge of safety and efficacy, which means it has to be big enough and there has to be a sufficient duration of follow-up. So the WHO and the FDA both agreed that the criteria for efficacy should be a 50% point estimate. And this is what you read about in the paper. These vaccines should reduce the incidence of SARS-CoV-2 or of COVID by 50%. But even more important that was the fact that there needed to be a 30% lower 95% confidence interval on that estimate. And what that meant is that we had very high confidence that however the trial was done, if our best guess is it might be at least 50% effective, we're sure that it will not be less than 30% effective. And this assures public health benefit. In addition, if it has this level of efficacy against COVID-19 in general, almost all vaccines are much more effective against severe disease than they are against mild disease. So this means it will be more effective, likely a greater effect on severe disease. It's likely that it will also prevent transmission. But the other thing that these statistical criteria did was they assured that clinical trials were large enough to assess other critical outcomes, including safety. And I can tell you that early on, plenty of manufacturers did not want to use a 30% lower bound. So many of them started and said, well, what if we just had a 0% lower bound that might prove that our vaccine was better than nothing? Wouldn't that be good enough in the middle of a pandemic? And yet, if you bring the first vaccine in and all you've done is prove that it's better than nothing, there's still a risk that it's not very good. And there's still a risk that it's not going to do what you need it to do. Because once you introduce a vaccine, it will probably cause people to be disinhibited, right? They're less likely to wear masks, more likely to take more risks. If you're evaluating many vaccines, I told you that when you evaluate many vaccines, there's a risk that one of them will meet the criteria due to a fluke. And so if you have a very relaxed criterion, that increases the chances that a vaccine will make it through without actually having any benefit or having very, very modest benefit. And by doing this, it reduces the risk that the first vaccine stymies development of later, better vaccines. Of course, a weakly effective vaccine is much less likely to induce herd immunity, which is an important concept in vaccination. And we really had one chance to get this right. And so the conclusion the WHO came to, and the FDA, was that a weakly effective vaccine could do more harm than good. And the kicker there was that a weakly effective vaccine also wouldn't have enough public health impact to have justified rushing it in the first place. So what this told us is that it was important to do this right and get the rigorous data that is needed to be sure that the vaccine is working, rather than to be sloppy about this. And so that is how this was done. And this sort of, this figure here, let me see if I have my little mouse here. This solid line shows you the likelihood of declaring success depending on what the vaccine's true efficacy is, depending on the lower confidence bound. So if the vaccine's true efficacy is 70%, regardless of the lower confidence bound, you have a pretty good chance of showing efficacy. But actually, if it's around 60%, if you have a lower confidence bound of 30%, which is what the FDA and WHO recommended, you actually have a better chance of showing that the vaccine will work because you have more people in the study. But the real problem is that if the vaccine has lower efficacy, maybe 20 or 30%, if you have confidence bounds that are either 0%, which is the triangle, or 20%, which is the circle, you start having unacceptable risks that a vaccine that doesn't work very well is going to make it through. And so this is the kind of thinking that with the help of the statisticians, we had to go through. And when we looked at this, we also asked the question, well, if we're going to assume how long the trial will enroll, what is being rigorous about this cost in terms of time? And the answer was that if we're doing several interim analyses, where early analyses would allow you to declare success if the vaccine was highly effective, the green dots here, or the green shaded areas, show the timing after starting a vaccine trial at which that it would take to show that a vaccine was effective based on different levels of efficacy of the vaccine. And you can see that even a 60% effective vaccine would have been shown to be effective within 5 months, based on a number of rather conservative assumptions that the WHO made early on. And what happened actually is that these trials enrolled patients much more quickly, and sadly, because many of these trials were being done in the US, where there was a lot of COVID, they all generated data even much more quickly than was expected here. And so it actually did not take long to get enough data to support this 30% lower bound. And it actually took much less time than was predicted. And then I think another major consideration here in terms of shortchanging the quality of the data is that if you get the results quickly, there may be other things you don't find, or if you do too small a study, you don't have enough people in the safety database. You don't have efficacy in subpopulations. You don't know how well it worked. And for instance, the AstraZeneca vaccine started with studies that were actually too small to show a 30% lower bound in the UK. And they then came out with data where they didn't have enough information to determine whether the vaccine worked in people over age 65, which then caused people in some countries of Europe, the regulators, to say, well, we don't want to give that vaccine to people over age 65. And so by being more rigorous and insisting on a larger and more complete study up front, that information became available. And you probably read in the news today that they obtained some data on subpopulations in a larger study that was done in the US, just reported that today. Also, this guarantees you have enough people to show efficacy against severe disease. It gives you a better chance of studying the potential for enhanced disease. And also, as long as the people are followed long enough in the study, then you can also start looking at the duration of efficacy. If you do only a very short-term study, you don't know how long vaccine-induced immunity is lasting. And what if it's only a few weeks? So you wouldn't want to base the deployment of a vaccine on very, very short efficacy data. And so the WHO group and the FDA did this as well, insisted that emergency authorization of vaccines should be based on at least 2 to 3 months of median follow-up. And you may remember back around October and September, the president was insisting that vaccines should be made available very, very quickly. And he was very upset when the FDA came out and said, well, no, we need to have at least 2 months of follow-up to be sure that these vaccines are working for adequate duration and that we also have adequate safety follow-up. But the FDA put its rationale both in a guidance document and actually also published its rationale, which I guess is not on this slide, but is on the next slide. But the ability to evaluate vaccine safety obviously depends on having a large enough study with enough COVID-19 infections to evaluate the potential for enhanced disease, as well as adequate length of follow-up. And here again is not a place where we want to take shortcuts because we're worried about vaccine hesitancy. We want the public to be confident that these vaccines have been thoroughly evaluated. So could less than 2 months of clinical trial follow-up reasonably support vaccine deployment? Well, the FDA thought the answer to this was no, both because confidence in vaccine requires rigorous evaluation, and that the FDA and WHO also both made the point that even after deployment, we'll still need more data. But based on experience with other vaccines, almost all vaccine-associated adverse events are detected within 2 months, but some within 6 weeks or so. And so shortchanging the follow-up might increase the chances that there would be undetected adverse events. And then there was also the concern that if the short efficacy follow-up was too short, it might allow deployment of vaccines with very short duration of efficacy. And this, even with 2 months of follow-up, would have been the fastest follow-up of any vaccine that has been made available in the US. So now that these trials have been done, what did they show? Well, here, this is the exciting part. Here's the Moderna vaccine in the upper left. This is the graph that they put together, where you can see they vaccinated at time 0. And so this is the vaccine group, the number of cases, and this is the placebo group, the number of cases. And you can see that these curves start diverging around day 12 to 14 after vaccination. And they found in their study 11 cases in the vaccine group and 185 in the placebo group. The Pfizer vaccine, very similar, also started diverging at around 12 days after vaccination. And they had 8 cases in their vaccine group and 162 in their placebo group. The Janssen vaccine started diverging a little bit later, and the number of cases in their vaccine group was higher, and yet this still represented a 66% overall efficacy, and it was over 70% in the US. Now, these vaccines were not compared head-to-head. And the Janssen vaccine was studied later and actually included testing under some pretty rigorous circumstances, including in Brazil, where there are new variants that are circulating that might have some resistance to the vaccine, as well as in South Africa, where it appears that some of their circulating variants also have some vaccine resistance. So the fact that the Janssen vaccine did this very well is actually a very good finding. And of course, the Janssen vaccine is a single dose. Now, a question that people might ask is, well, if we start seeing that the vaccine is effective as soon as 12 days after vaccination, but for Moderna, the second dose of vaccine is given at 21 days, and for Pfizer, the second dose of vaccine is given at 28 days, maybe we don't need the second dose. And yet, all of these people in these studies had the second dose, essentially all of them do. And so this is what you find when you get the second dose of these vaccines. So while there's probably some protection after the first dose, and that's very encouraging, it doesn't mean that you can get away without the second dose. The adverse events have been generally very tolerable, and I just put on this slide those reported for mRNA vaccines, really injection site reactions, the main one, fever, headache, muscle pain, joint pain, and fatigue. And many of you who've been vaccinated may have experienced one or more of these side effects, especially after the second dose. The older you are, and I'm sorry, since I'm imagining most of you in the class are in the younger group, if you've been vaccinated, you're probably more likely to have gotten these side effects than people who are over age 65. And there is some risk of allergic reactions after these vaccines as well. It was reported initially and appeared to be quite high, but with additional data, it's turned out that it's not really much different from other vaccines. But it was just that people were being very vigilant at the time, which then led to a lot of reporting and perhaps reporting of events that might have been allergic or people thought were allergic, but turned out not to be. What about other considerations for these vaccines? Well, the Pfizer vaccine is actually authorized down to age 16 because they included 16 and 17-year-old participants in their clinical trials, while Moderna and Janssen vaccines are above age 18. The Janssen vaccine can be stored in a regular refrigerator, whereas the Moderna vaccine needs to be stored in a regular freezer. And the Pfizer vaccine, at least initially, needed to be stored in an ultra low temperature freezer. but both the Moderna and Pfizer vaccines can be stored at room temperature for 6 hours after puncturing the vial. These are all multi-dose vials, which gives you some time to give multiple doses. The volume of these doses is a little bit different. The total amount of dose is different, but the Pfizer and Moderna, as I said, are 2-dose vaccines. The Janssen is a single-dose vaccine. And Janssen is hoping by the end of 2021 to have 1 billion doses, or actually courses, 1 billion doses. Pfizer is hoping to have 2 billion doses, or enough to immunize 1 billion people. Moderna, a little bit less. But of course, if you put this together with a worldwide population of 8 billion people, and even if you add what is being made available from other vaccine manufacturers where the vaccines are not yet authorized in the US, what you have is still a very big vaccine shortage around the world. So the WHO has stated pretty strongly that more vaccines are needed. And of course, although these early vaccines are really have been home runs in terms of what they have been able to produce, even vaccines with lower efficacy against mild disease, as long as they protect against severe disease, still can have an important role to play if they have other advantages, including in deployability, cost, the number of doses that need to be given, single-dose vaccines obviously being preferred for deploying in hard-to-reach places, safety or duration of efficacy, which is something we don't really know for any of these vaccines. And the most efficient way to evaluate these will be placebo-controlled trials. Even now, although we can talk about the circumstances under which it's okay to use placebos in vaccine trials. You can compare vaccines against an active comparator that is known to be effective, although it takes a lot longer to come up with the results. And there are other limitations as well, including if the types of viruses that are infecting people change. For instance, if the variants take hold after the first vaccine was evaluated, we might not know that that vaccine was effective when we compare a new vaccine against it. So the FDA has made these vaccines available under emergency use authorization in the US. I just want to say a word about that versus licensure. Under EUA, the benefits need to outweigh the risks, but the FDA took a very stringent view of this and insisted that this be based on data from at least one well-designed phase 3 trial that demonstrated the vaccine's safety and efficacy in a clear and compelling manner. And so that's a little bit of a different standard than was used by the FDA for some of the drugs that have been authorized for COVID. But for vaccines, we felt that was very important because they'd be given to healthy people. And then for licensure, of course, which none of these vaccines are yet licensed, but we'll have the standard is substantial evidence of efficacy and safety. And there will be more data available at that point, as well as longer-term efficacy and safety data. So what does responsible deployment of these vaccines look like in emergency? Well, we want to get the vaccine to people who need it, but it's critical to remember that there's a lot we still don't know about these vaccines, and we would like to understand what is happening. So it's going to be important to collect additional data, both from placebo recipients as well as from observational studies. And but there are circumstances under which placebo-controlled trials can still be done. And of course, this additional study will be especially important to address vaccine effectiveness against emerging variants. Well, what about these variants? The virus has been evolving from the beginning. In fact, we believe it's been evolving from an animal virus before it made the jump to humans. And natural selection as these viruses evolve tends to allow viruses with increased transmissibility to emerge. So the risk is that over time, if it's possible for the virus to become more transmissible, it probably will, which means that it will spread more rapidly. Now, a hope is that as it does that, it might also become less severe, but so far we have not seen any loss in severity of infection. And yet some of these new variants do have increased transmissibility. And some of these variants are even able to evade aspects of the immune response. But so far, none of them have been seen to evade vaccine-induced protection against severe disease. However, WHO and others are following the situation very closely to see if and when new vaccines might be needed. This might be as boosters for people who've already been vaccinated, or to come up with whole new vaccines that might provide broader protection to people who haven't yet been vaccinated, including potentially people who've been previously infected. And then to define what potential antigens that new vaccines should protect against. FDA and other regulators have also been thinking about that and have already defined and published in a guidance document methods for evaluating new vaccines in the event that they're needed. And this is based on how can we do this quickly so that we can have confidence in these new vaccines while also not allowing variants, should they start evading vaccine-induced immunity, to bring us back to square one. But these are all precautionary measures and shouldn't reduce confidence in existing vaccines, which clearly save lives. So for vaccines, our key information gap is how long will vaccines protect recipients, either against the original virus or evolving variants? We don't know that yet, and yet this is being very closely evaluated. What can we say with confidence? Well, they do protect against serious disease. They'll reduce hospitalizations and will likely reduce transmission of the virus. They'll work best as part of an integrated public health strategy, which includes continued attention to distancing and mask use. And a deployment of vaccines now, along with these public health measures, will reduce the likelihood that dangerous variants emerge. So to conclude, I've given you sort of a whirlwind tour here of a lot of things, but this is how we've gotten to where we are. It's really been a story of worldwide collaboration, and it remains the logical path forward. A lot has been accomplished, and there's also still much to do. And we can still look at the US numbers and see that while since January, COVID cases have started coming down, we sort of have been in a plateau for the last couple of weeks. And there's some states where they're actually rising again. And of course, the observed high efficacy of vaccines provides hope we can soon bring life back to normal as we get people vaccinated, either with the help of these vaccines or others that induce similar immune responses. And actually, what I'd hoped to do was do the question before I gave the talk, but I jumped into it before I asked you to do that. So we have a little poll which will sort of give you a flavor of the kinds of things that we in the public health arena think about when we're working in this area. So I don't know, Jen, if you want to put the question up and give the students a chance to answer.
Yeah, so the poll is up right now. It's, what do you think is the most important COVID vaccine related priority? The options are speeding up vaccine availability in the US, making more vaccine available for the rest of the world, making vaccines available to children, getting and providing more information on existing vaccines, addressing vaccine hesitancy in some other way, or something else entirely. So go ahead and take a few seconds to respond to that poll. We have over 90% of responses in. Should we share this now, or do you want to wait until later?
Well, let's go ahead and share it now.
Okay, great. So yeah, it looks like 37. Do you want to do this, or you want me to read off the results?
I know, well, I can read it too, I guess, since I can see it here.
Yeah.
So most of you would like to speed up vaccine availability in the US. Almost equal number would like to make more vaccine available for the rest of the world. Making vaccines available to children seems like a low priority, which is actually interesting because there's an awful lot of talk in the newspapers about the importance of getting vaccines to children so that schools can fully reopen in the fall. And everybody talks about the harm to children of not having full-time hours in school. And yet making vaccines available to children is maybe one of the things that will persuade the teachers that they're not at such high risk. Getting and providing more information on the existing vaccines doesn't seem like such a high priority. It sounds like you all know enough, at least enough to know that you would like the vaccine availability sped up. And addressing vaccine hesitancy in some other way is, I think, also important to some of you. And it may be that if you add those two together, this question of vaccine hesitancy is a significant one still. You know, what information can we get that will persuade people that the vaccine is safe and effective? And then 3%, 5 people said something else. So I'll be very interested in hearing what the something else is when we get to the questions. But, you know, of course, these are all priorities. Making vaccines available in the US, a lot of this has to do with looking at manufacturing facilities, which I didn't talk a lot about. But the FDA is in close contact with these manufacturers and figuring out how can they increase the throughput? How can they make more vaccines faster? Some of this is also, of course, the availability of the reagents that are needed to manufacture these vaccines, because they're made with complicated components. And so that may limit this. But of course, what the FDA wants to do is make sure that whatever vaccine is made available is vaccine that people can have confidence in. And so there's a very big part of what the FDA does is actually based on this notion of studying the manufacturing and the testing of vaccines. Because you can have a clinical trial that shows that a vaccine works, and that's great. But what you've shown then is the stuff that you studied in that clinical trial works and was safe. But how can you be sure then that the vaccine that you're going to make after that for many, many years is going to be the same as that which was in the clinical trials? And the only way to do that is by having a very well and thoroughly characterized manufacturing process where the product is being tested and aspects of that process have been tested as it's been going on. And then the locations where the vaccine is being made, where they're trying to implement that process, have also been very carefully investigated and inspected to make sure that this is all being done correctly and properly in a way that people can continue to have confidence in the vaccine. So the FDA's role, of course, is to try to address the scientific end of confidence in vaccines and vaccine hesitancy. We're at the FDA, we're not so much in the business of trying to persuade people, oh, it's good for you to take this vaccine. That's the CDC's job, is to make recommendations about deployment. What we want to do is make sure that the testing of the vaccine has been complete and that the information that has been put out about the vaccine is accurate. And by sort of staying out of some of these more subjective questions, we hope then that we earn the trust of the American people, because they know that we can be counted on to objectively look at the science. And so we tend to be very science-based in this. So it's interesting, there's this whole notion of vaccine nationalism, which comes up, and you've probably read about that in the paper too, where the WHO especially is very concerned that the US, for instance, and the UK, and in fact, even the rest of the European Union, now that the UK is not a part of that, see that there are more vaccines per capita in certain countries where the US and the UK are doing very well. Israel is doing quite well too, due to some smart deals that they made. But there are parts of the world where vaccine supply is very, very low. And these are places which also are where people are at risk from COVID. And so there is perhaps a question of fundamental fairness of whether every American should be vaccinated, for instance, before anybody is vaccinated in a country that has not been able to put as much money into vaccine development. And of course, we're all a part of a larger world. And what we've seen is that these variants can develop anywhere. And so if you only were to vaccinate Americans and not vaccinate people in other parts of the world, you may well have a worse long-term outcome than if you were also to make vaccine available elsewhere. So there is this fundamental tension there. And so it's interesting to see you all split pretty much down the middle on that. Okay.
That's great. Oh, go ahead.
Yep. So why don't we go to some questions?
Okay, great. I have a few clarifying questions, but since you bring up other countries and getting vaccines out to other countries, one of the questions, I'll come back to the clarifying questions in a second. Students were wondering if you could just kind of talk about what it looks like trying to get the vaccine to places that don't have the sort of healthcare infrastructure, or really infrastructure, right? Like I work in Madagascar and Uganda. Those are places where keeping things cold is very difficult. So what are the challenges with that? And do you have any thoughts on how we could better facilitate making sure that other people are also getting the vaccine?
Yeah, so the cold chain is obviously a big issue, where if you really need to keep the vaccine cold, it's much harder than even if the vaccine needs to be kept at a refrigerator temperature, because you'll find refrigerators almost anywhere. But there are some incredibly impressive examples of vaccination campaigns that have been carried out under very difficult circumstances. And the WHO actually, with volunteers essentially who came only from Africa, because people who are not from Africa were probably at significant risk. In the Democratic Republic of the Congo, were able to deploy very large numbers of doses of an Ebola vaccine enough to get the outbreak there under control, hundreds of thousands of doses. And this is a vaccine that needs to be kept at ultra low temperatures as well, at least for most of the time. And so they were able to use the vaccine on ice for short periods of time, but really had to think about how they were deploying that. And this is something that was done by Africans for Africans with some logistical help from the WHO. So we are getting better at that, I would say. Obviously, polio vaccine has been deployed around the world. Smallpox vaccine was deployed around the world. There's an incredible success story about a vaccine that is called Menafravac, which is a meningitis vaccine that was developed for Africa to be given in the meningitis belt of Africa, where this would be an annual event where large numbers of people would get meningitis and die from it. And so there was this decision, and this was actually run by one of the nonprofits I talked about, at least it was on my slide, where they developed a vaccine. They licensed very inexpensive technology. It was produced in very cheap plants, and they were able to deliver this vaccine across the meningitis belt in such a way that this annual meningitis event is almost non-existent now. And so there are huge vaccine deployment successes. The other thing that my friends at the WHO always remind me about is that in Africa, people don't have the luxury to have vaccine hesitancy. So they know that the vaccines are going to protect them. And so if for instance, when they were deploying the Ebola vaccine to people who they thought were at risk, who might have been a contact or a contact of an Ebola case, they would get in the high 90%, 99 or so percent acceptance of the vaccine. You did not have people who said, "Oh, I think I can do without this vaccine," and so it seems as though, at least in these settings, people in the developing world are able to cut through some of this, and in many cases are more accepting of vaccines than we are here in the U.S. and in the developed world.
I think that's a great point. I also like how you talk about, the need to be able to address this issue in different places, because a lot of the students had questions about why are companies and the government continuing to pursue other types of vaccines when we have 3 already available to us here? Like, should everyone be putting all of their focus in making sure that they are producing enough of the ones that we already have approved rather than continuing to explore these other options? And I feel like what you just mentioned kind of gets at that what we have available right now doesn't necessarily work great long-term in every situation. But maybe you could just talk a little more about that too, in terms of where should our focus be and really making sure we have enough supply of the ones that have already been approved, or in continuing to see if there are other options that might be even better long-term.
Of course, we're not going to know. There'll be a big question if there are variants that start evading vaccine-induced immunity, what will be the best approach? And will we need would you be better off with a booster of a different vaccine from the one you started with versus a booster of the same vaccine from what you started with? And these are the kinds of data that people are going to need to generate. But these other vaccines are essential for the rest of the world. And so some of these are being made by companies in the US and some are being made by companies outside of the US. In general, if they're evaluated in the US, this provides some extra cachet and valuable data. For instance, although some of these trials initially were done in a way that was perhaps not large enough to yield rigorous results and left some questions about these vaccines, the fact that in the US we insisted that these vaccines be evaluated rigorously is generating data that will be useful in the rest of the world also. And so I think that if we look at this solely from a US perspective, we probably do have enough vaccines for right now. But of course, we don't know what we're going to need a year from now. And so I think that given all of the unknowns, it's probably a little bit early to say we're going to stop evaluating vaccines.
Yep, That's a great point. So let me go back to some of these clarifying questions. You talked about the different vaccine technologies, and you had both mRNA and modified mRNA vaccines on that list. Can you explain the difference between those?
Sure. This is actually the genius of the mRNA vaccines that have been successful so far. And that is that when a virus infects a cell, the cell notices that it's been infected. And one of the ways it notices that it's been infected is it notices that RNAs are being made that it doesn't expect to be being made. It might notice that there's some foreign DNA in the cell. And what the cell does then is it creates an interferon response. And what that interferon response does in that cell is a lot of things, but it sets up a protective response in the cell to shut down the virus that has infected the cell. So this is one of our primal defenses against viruses. But it turns out that if you inject mRNA into a cell, the cell thinks it's a virus and it does the same thing. And so it tries to shut down what that mRNA is doing. And in so doing, it reduces the amount of protein that can be made from that mRNA. So the genius behind these mRNA vaccines that have been successful so far is they are mRNAs that have a modified backbone. And for those of you who know the biology, instead of having a uridine in the RNA, they have a pseudouridine. And a pseudouridine is found in tRNAs and in RNAs that sit in the cell that the cell needs for its own purposes. And therefore, by making the RNA look like an RNA that belongs in the cell, it defeats this interferon response. The cell no longer recognizes the RNA and thinks it's a foreign RNA. It realizes or it thinks it's an RNA that belongs there. And it doesn't induce that interferon response, which then allows more of the protein to be made. Now, there's another trade-off there, which is that if there's no interferon response, then you also might not get enough of an immune response. And so I think there is still some weak interferon response from some of these modified mRNA vaccines. And also there are other ways in which these products induce inflammatory responses, including, and I think the lipids that go in with them that are used to bring them into cells also increase the local inflammatory response that allow you to get the best of both worlds. You get lots of the protein made, but then you also get enough of an inflammatory response that you get immunity. So it's not just a question of taking mRNA and putting that into cells, because that actually doesn't work very well. Although there are some companies that are still looking at that and trying to find other ways to tamp down those kinds of interferon responses and allow for the protein to be made, besides modifying the backbone of the mRNA. And I also mentioned self-amplifying RNAs. And that's another idea where people take RNA and they include parts of a virus that is actually able to make more copies of that RNA once it gets into a cell. And then by making more copies of the RNA, that in theory will increase the amount of protein that can be made from that. The downside of that is that the RNA that is made from that looks like RNA that is made from a virus. And so the cellular defenses against a foreign virus could kick in. And so to make that work, then you might need to do other things to try and defeat those defenses as well. So the bottom line is there's a lot of science behind all of these things. And a lot of this comes out of many, many years of work, understand how it is that the cell and the immune system react to different things. But these mRNA vaccines and the very successful ones wouldn't have been possible without this observation that you can defeat this interferon response by using pseudouridine instead of uridine, or by changing the backbone of the RNA. So that's probably too technical an answer, but.
No, I think that's great. I mean, and I really appreciated at the beginning of your talk when you were saying how important continual efforts, doing bench science help to be able to make this go very quickly once we have something that we need to respond to.
No, and in fact, of course, all of these RNA platforms were already being studied for different pathogens. And so there was already an understanding of how these RNAs were getting into cells, what types of immune responses they might generate, how safe they would likely be, which also allowed these platforms to move fairly quickly into clinical trials.
That's great. So one more clarifying question. Some students were wondering then, the way that they tested the vaccines, did people actually have to be exposed to the virus in order to test when they were doing the trials before approval by the FDA, did people actually have to be exposed to COVID?
Sure, yes, but not deliberately. So what you do is you look for volunteers for a trial, and you give, in most cases, half of them, but sometimes it's a different ratio, the placebo, and give half of them the vaccine. And then you actually remind people that they should be careful. And this has been a tenet of HIV vaccine trials, where HIV is often transmitted sexually, of course, and yet part of your responsibility as somebody who's conducting an HIV vaccine trial is to remind people that they should be using protections that reduce their acquisition of HIV. But in spite of those kinds of reminders and recommendations that people avoid exposures, people will get exposed anyway in the community and inadvertently. And so what you do is you then compare the number of cases that arose in the placebo group with the number that arose in the vaccine group. And then you use that to calculate that if the vaccine group had been like the placebo, by how much did you reduce the total number of cases? And usually it isn't an exact division of one number by the other, because there might be slightly different lengths to follow up in one group versus another, slightly different numbers of people, but that sort of gives you a rough answer. Now, there has been this discussion of human challenge studies where there's been this idea to deliberately expose people to the virus. And indeed, there are many people who said they would volunteer for such studies. Of course, if somebody gets into a study like that and they're deliberately exposed to the virus, there's some risk for them. And so one of the things we want to do in clinical trials is avoid doing harm to the people who join the trials. And so one of the things that, on the other hand, I guess what I'll say is, there are people who said that if you could do such trials, maybe you could get certain kinds of answers more rapidly than you would otherwise get them. But I think the concern about making people sick has been a strong one. And so until there are available very good drugs for treating COVID, so that if somebody started getting sick in such a trial, you could, in essence, rescue them. I think there was a sense that one ought to hold back on that. And so that, together with the fact that when people have looked at this very carefully, with so much wild circulation of the virus in the community, even now, there have been very few questions that you could imagine answering using a human challenge study that couldn't be answered using a wild exposure of people who were in a regular clinical trial. These human challenge studies also have to be undertaken very carefully. So it's not like you just, inject somebody with a virus that you've gotten from wherever. If you're going to do a human challenge study, somebody has to make a challenge stock and they have to characterize it. Make sure you know what virus you're giving to these people. You have to make sure that that challenge stock isn't contaminated with something else. And you have to make sure that it really is what you think it is. Because what if it mutated while you were making it and it's different from what you thought it was? And so it's not so simple. So you can't just say, I'm going to do a human challenge study tomorrow. It generally takes a few months to even get ready to do it. And the question is, in those few months, maybe you could have already gotten the answer in some other way. So these are the reasons why there's been not so much enthusiasm for those trials. And yet there are some who are very enthusiastic about them and continue to advocate for them. And as I mentioned earlier, there are some such trials that people are proposing to do in England right now. Although it's still not clear that those will yield answers that can't be gotten in other ways. Yeah,
that sounds like a very stressful way to test the vaccine. So on that note, a lot of students have had this question about if COVID vaccines are going to go the way of flu vaccines, and every year there will be a new type of vaccine developed that everybody, who wants to gets it each year. What do you think that looks like moving forward? What's going to happen with the COVID vaccine?
We don't know. It might be that we'll need more vaccines even sooner than a year, or it may be that the rate at which it mutates is less than every year. So it's probable that this virus is going to keep mutating. But of course, we all worry about this virus because of its ability to cause serious illness and send people to the hospital and to kill people. And so if it may be that the current vaccines, even if they allow people to get infected at some point as the virus mutates, will still protect against serious disease. And then the question will be, well, would we really want to introduce new vaccines if they're not providing substantially greater protection against serious illness? I think our goal as a society needs to be not to make COVID go away, because it probably won't go away completely, but we want to knock it down to where it's a disease that is manageable. And so if we can knock it down to a common cold from where it is now, where it is causing the disruption to health, and of course also economic disruption that it's been causing, that will probably be good enough. So I think it's probable that the vaccine will evolve to evade immunity from the vaccines over time. I don't know how long it will take, and the question will be, in the end, how significant are the public health consequences of that? And that's, I think, what is going to drive the timing of either booster doses or vaccines that are addressing these new variant strains. But for people who are afraid that this is going to happen and say, oh, I'm going to wait with getting my vaccine because probably there'll be a new one soon anyway, I would advise against that, because the vaccine that you can get right now is the one that will protect you right now. It's also the one that will help protect everybody else, because the more people who get vaccinated, the sooner we can put this behind us, the sooner businesses can open, the sooner the case counts go down. And so I think what people don't understand is that they see this as an individual issue, when in fact it's just as much a societal issue. If we can't open up our society in which people continue to be suspicious of each other and afraid to do things that they would like to do, then we can't move forward. But the way we get there is both with vaccination, and even if people have been vaccinated, it still is sensible for them to wear masks and to continue to distance themselves. And the reason is because people who've been vaccinated are not 100% protected. They could still transmit disease to somebody else. And so it's not a perfect thing. Another reason is because these new variants might increase the chances that they get infected asymptomatically even and could transmit to somebody else. So it's still another reason to continue to be careful, because we won't know when to flip a switch when that starts happening. And then the other thing, of course, is the practical one, is in a society where the proportion of people who are vaccinated is still very small. If you tell people they should stop wearing masks when they're vaccinated, there may be plenty of people who prefer to wear masks, not to wear masks, who haven't been vaccinated. They might just claim they're vaccinated. And then society will lose a lot, because that will then greatly increase the spread of the virus. So for all of these reasons, for individual and societal good, I think it's important for people to view this as a combined public health strategy, where they don't give up on the vaccines, don't delay the vaccines, and also don't give up on the public health measures.
That's such an important message. Thank you for saying that. So related to that, I mean, I think, as you said, we don't know yet in the future if an additional change to the vaccine will have to be developed, or how frequently you'll have to get a dose of the vaccine. But students are wondering if you can talk a little bit about, at least right now, with the rollout that's happening, what is this idea of herd immunity, and what numbers are we trying to get to reach some level where we do actually see our society starting to open back up safely?
So, herd immunity is not an all or nothing thing. And so if you look at epidemiology textbooks, they say, oh, 80%, or for measles, 90%, whatever. And this gets us to where we need to be. But and I may be in the minority here, so I'll preface this by saying that this is a personal and not an official opinion. But it was very interesting. I worked on the chickenpox vaccine in the 1990s, and even before a significant number of people, or a large number of people had been immunized, you could start seeing herd immunity. And the reason was because chickenpox tended to travel in families. And so if one or two kids in a family get immunized, that would protect the other kids in that very family. And so I think we actually start seeing herd immunity, possibly even with the level of previous infection and vaccination that has now occurred. And I think that this is already probably starting to protect people. But of course, it's protecting people in the context of current behavior. So if people continue to distance as they have done since January, what we've seen is that the so-called R naught of the virus has been below 1 and the total number of cases has gone down. And the vaccines may have played some role in that, but probably not much, because the case count started going down before the vaccines were available, which meant that we actually, in the context of the distancing that was being done at that point, the mask wearing and distancing, we had enough herd immunity that the total case counts were starting to come down. I guess to me, the thing about herd immunity is that the more people who are protected in one way or another and are protecting other people in one way or another, the better off we are. And so it may well be that even with, well, certainly well under half the population infected, and also not that many people relative to the entire population vaccinated yet, in the context of everything else we're doing, that may be enough herd immunity to be making a difference. And so that's another reason why we sort of don't just say that you would get vaccinated and stop everything else, or that we would stop everything else once we reach a certain level of vaccination. But I think we need to continue to be careful. Now, what does this mean for businesses that want to open up? Well, of course, to me, the simplest thing is that everybody can always wear a mask. And so if one's goal is to keep people in one's community employed and keep people healthy around one, it certainly will always be sensible to wear a mask until we're well through this. The question then becomes, how many people can you allow into a store at once? How many can be in a restaurant at once? What I think is that as summer approaches, people will be more and more comfortable with outdoor dining, and so the need to do indoor dining may start declining as we move forward, and so that may be less of an issue. But I'm not ready to give a specific number which we have to reach, because what I see is that the more people who are in essence participating in blocking the spread of this virus, the better that is for everybody, even now, based on the data we have right in front of us.
That's totally fair. Yeah. So students are also wondering, they have, family members and friends who are hesitant about getting the vaccine. We only have about a minute left, but I guess I just was hoping you could address, like, some of the specific questions are, have people died from the vaccine? Can you get the vaccine if you're on other medicines? Can you just address kind of directly the safety of these vaccines?
Yeah, so there aren't any other medicines that should prevent you from getting the vaccine. There are some people who died after getting the vaccine, but there's no evidence that anyone has died from the vaccine. There have been some serious cases of allergic reactions, including anaphylaxis, which people consider possibly fatal. But because these vaccines are being given in a setting where that can be treated, to my knowledge, nobody has died from that. It's all been appropriately treated. And so these vaccines, certainly relative to the risk of getting COVID, are very, very safe. And this is what, hopefully some of the slides I went through help to persuade your students of, is that this has been very rigorously looked at. And this has been looked at with the idea, not just, oh, this is probably better than nothing. This has been looked at with the idea is, we're going to be very rigorously sure that if you're faced with a decision to get this vaccine, that people will be better off getting it. And so we do have enough efficacy and safety data. Now you can add to the efficacy data that was in the clinical trials, the many millions of people who've gotten the vaccine were being followed up in various ways by the CDC and the FDA, as well as many more millions who've gotten vaccines in Europe and other parts of the world who are being followed up with surprisingly few side effects when you consider that bad things happen to people every day. And so the chances that something bad will happen to somebody on the day that they get a vaccine are very, very high. And so far, we're not really seeing greater incidence of any of these kinds of things after vaccination than is normally expected.
That's great. Well, the students have so many questions. We could talk to you all day, but I know that you have very important things to do. One thing that I've seen over and over again in the Q&A box from the students is a message of thank you to you. And I do want to emphasize, we really appreciate you not only being here and talking to us, but also the work that you are doing right now. I mean, we talk in this class a lot about how incredible it is that we have a vaccine already that's being distributed to people. We have multiple vaccines already being distributed to people. So thank you so much for your time. And also thank you for everything that you're doing.
Well, thanks. This is a huge amount of teamwork. The number of people around the world who pulled together to try to make something work here has been incredible. And it is really gratifying that people are doing this, and maybe not in all cases completely thrusting aside their egos and their self-interest, and yet enough that we can make incredible progress. And this wouldn't be possible without people really approaching this with a sense of altruism and a desire to help people. And so it's a real thrill to be working with these people as well.
I love that. Thank you so much. Thank you for being with us.
My pleasure.
Thank you.
Thanks. And good luck to all of your students.
Thank you. So we will be back here on Wednesday. Until then, stay safe.